Welcome to another Explainer. I'm so glad you're joining us today. Now, we're going to put on our clinical geneticist hats for a bit and look at the absolute foundation of human health and disease. We're talking about mutations. I want you to imagine the human genome as this incredibly vast, intricate blueprint. We're talking about a genetic code that contains 3.2 billion base pairs. It's just staggering. Now, every time our cells divide, they have to copy that entire blueprint perfectly. But as we know, nature isn't flawless, right? Sometimes microscopic typos occur within those 3.2 billion letters. And these typos, well, these are mutations. Understanding exactly how they happen is basically the key to everything, from diagnosing ultra-rare diseases to unlocking the entire future of gene therapy. So to kick things off, let's do a little clinical quiz review. We've got two very well-known genetic conditions, cystic fibrosis and Down syndrome. Both of these fundamentally alter human health, but on a molecular level, they represent entirely different scales of genetic error. So can you guess the structural difference between the actual mutations that cause them? Take a second, lock in your answer, and then we'll unpack the biology behind all of this.
Okay, to answer that, we obviously need to define what a mutation actually is first. Fundamentally, it's just an alteration in the nucleic acid sequence of a genome, but the analogy here is what's really interesting. If our DNA is a massive text document, think of it like a giant library of encyclopedias. A mutation isn't just one type of mistake. It could be a single deleted letter on page 400, or, actually, it could be an entirely duplicated chapter, or even a whole book printed backwards. These changes usually stem from replication errors when cells divide, or from environmental DNA damage, like UV radiation, that somehow manages to slip right past our cellular repair mechanisms.
And knowing how these errors happen brilliantly illustrates when and where they actually matter in a human lifespan. On one hand, we have germline mutations. These originate in the parental gametes, so the sperm or the egg. Because they are literally baked in from day one as the embryo develops, that exact mutation is copied into every single cell of the resulting person's body. These are the heritable mutations, passed down through families. On the other hand, we have somatic mutations. These happen after conception, during the countless cell divisions of your life. A somatic mutation localizes to just one specific organ or tissue type, so it won't be passed to your children, but this specific type of genomic instability is heavily linked to aging and the development of localized cancers.
So the absolutely crucial point here is that genetic errors come in vastly different sizes. Medical students are usually taught to categorize these broadly into small-scale and large-scale changes. First, you have point mutations. These are your surgical single nucleotide changes, literally just a single letter swapped, added, or removed. Then, on the complete opposite end of the spectrum, you have chromosomal abnormalities. These are massive, large-scale structural shifts involving entirely missing or extra chromosomes. It's essentially adding or deleting entire volumes of our genetic encyclopedia.
Right, let's move back to our quiz and see how this builds our understanding. Remember the first part about cystic fibrosis? Well, cystic fibrosis is caused by a point mutation. Specifically, it's often this tiny deletion of just a few nucleotides on chromosome 7q that affects the CFTR gene. From a clinical perspective, it is just staggering to think about a family's journey here. Moving from noticing a child's first symptoms, navigating the extreme anxiety of genetic testing, to finally receiving a diagnosis that traces massive, systemic, respiratory, and digestive issues, all the way back to a single missing letter in a sequence of billions. It's wild.
But, not all point mutations are created equal. A single altered base pair can have vastly different effects. A synonymous or silent mutation changes a letter, but because our genetic code has built-in redundancies, it still produces the exact same amino acid. So, no harm, no foul. A missense mutation, however, swaps out an amino acid entirely. This is actually what causes sickle cell disease. One wrong amino acid fundamentally changes the shape of a red blood cell. Then you have a nonsense mutation, which is even more abrupt. It creates a premature stop codon, basically telling the cell to just halt building the protein halfway through. And a frame shift, that disrupts the entire reading frame, scrambling literally everything downstream. Identifying exactly which type of point mutation a patient has is vital today because modern gene therapy research is increasingly allowing doctors to choose targeted treatments that address that specific mechanical failure.
Now let's look at a very specific and frankly fascinating type of mutation, the trinucleotide repeat. Consider the biological model of a really devastating neurodegenerative disease called Friedreich's ataxia. Imagine a specific three-letter sequence in the FXN gene, the letters GAA. In a normal chromosome, this GAA sequence stutters or repeats fewer than 40 times. But in patients with Friedreich's ataxia, this sequence expands massively, jumping to over 600 or even 900 repeats. And the consequence of this is huge. All those extra repeats cause the DNA structure to physically clump up and condense into something called heterochromatin.
Let's break down that exact biological mechanism step by step, because it's important. First, that GAA repeat expansion occurs. Second, because of the sheer physical bulk of that repeating code, the DNA condenses tightly into that heterochromatin we just talked about. Third, because the DNA is so tightly clumped, the cellular machinery simply cannot read it. This is called promoter silencing. And finally, step 4. Because the gene can't be read, the body stops producing a vital mitochondrial protein called fracticaxin. This microscopic stutter leads directly to the loss of coordination, muscle weakness, and cardiac issues that patients face every single day. It's a perfect, albeit tragic, illustration of how a structural DNA anomaly disrupts normal cellular function.
Okay, let's jump back to the second half of our initial quiz, Down syndrome. If cystic fibrosis is a surgical point mutation, what on earth is Down syndrome? Well, it falls right under chromosomal abnormalities. Down syndrome is definitely not a single typo. It's a massive structural shift known as trisomy 21, which means the patient has inherited an entire extra third copy of chromosome 21.
These chromosomal abnormalities involve large-scale changes that drastically alter the dosage of hundreds of genes all at once. You can have duplications, like the trisomy we just mentioned. You can have deletions, where huge regions of a chromosome are simply lost entirely. Translocations happen when pieces of non-matching chromosomes break off and basically swap places. And inversions occur when a segment of a chromosome breaks, flips completely backwards, and reattaches itself. We are talking massive architectural shifts in the genetic blueprint here.
So a natural question is, does the size of the mutation dictate how common the disease is? Well, let's look at the prevalence. We have cystic fibrosis, a tiny point mutation, affecting about 1 in 100,000 people. Friedreich's ataxia, a trinucleotide repeat, affects 1 in 50,000. And Down syndrome, a massive whole chromosome duplication, is actually much more common, affecting about 1 in 1,000 births. What's really interesting here is that the physical scale of the genetic error absolutely does not linearly correlate to how prevalent the condition is in the population.
Let's expand on that population impact for a second. To put things into perspective, think about a complex genetic condition like Alzheimer's disease, affecting about 1 in 177 people. Then you have cystic fibrosis at 1 in 100,000. But then look at aceruloplasminemia, affecting just 1 in 2 million people. This vast spectrum highlights exactly why global tracking and massive data sharing are so absolutely essential for rare disease research. For a doctor to even begin to understand a mutation that occurs once in 2 million people, they need data from all over the world. And for the families managing these chronic, ultra-rare genetic conditions, finding support resources and building community awareness can feel incredibly isolating. It really makes early detection and public awareness absolutely vital.
But why are some of these diseases so incredibly rare in the first place? It largely comes down to inheritance patterns. We each inherit 2 copies of every gene, right? One maternal, one paternal. If you have a heterozygous pattern, only one allele is mutated. Now, for dominant conditions, that's enough to cause the disease. But many rare genetic disorders like cystic fibrosis or Friedreich's ataxia are recessive. This means they require a homozygous pattern, identical mutations on both the mother's and the father's allele, or a compound heterozygous pattern where you inherit 2 different mutations on the exact same gene. You basically have to inherit 2 specific molecular typos at the exact same location, which, mathematically speaking, is just way less likely to happen.
Up to this point, we've really looked at mutations purely through a medical lens, as drivers of disease and suffering. But I want to pivot for a moment and show you the bigger picture, beautifully captured in this thought, based on Moto Kimura's evolutionary theories. Gene duplications and point mutations actually provide the raw material for human evolution. When a gene duplicates, the original copy can just keep doing its vital job, while the extra copy is suddenly free to accumulate mutations over millions of years. It can make evolutionary experiments. Without these genetic typos, life would never adapt, change, or evolve. We literally wouldn't be here without them.
Which leaves us with this final, incredibly exciting thought. We have spent millions of years completely at the mercy of these random typos in our genetic text. But today, through genetic testing, we can pinpoint the exact letter that went wrong. And with advancements in gene therapy, we are standing on the precipice of being able to correct those specific mutation types. So, how will our growing ability to intentionally edit our own biological text reshape the future of human disease, aging, and evolution itself? It is a profound question, and for sure, one that makes the study of genetics the most thrilling frontier in modern medicine. Thank you so much for joining me for this explainer. Keep asking questions, and I'll catch you next time.